Enteric viruses are mostly found in different aquatic reservoirs, including groundwater, sewage, streams, rivers, and marine water. They are associated with health risk and are responsible for infections in humans. Enteric viruses commonly cause gastroenteritis and are mostly transmitted through the fecal-oral route. Other waterborne diseases cause acute hepatitis by hepatitis A virus and hepatitis E viruses, with significant associated mortality, especially regarding hepatitis E in pregnant women. Actual protocols for monitoring water quality proposed for humanitarian contexts have been designed taking bacteria such E. coli as indicator of fecal contamination. But it is already known that there is no correlation with other microorganisms as viruses that are more resistant to many inactivation processes. Until now, detection of viruses in humanitarian crisis settings have been performed by adapting existing protocols to the conditions of those contexts and shipping samples to reference laboratories for molecular detection of viruses.
There is a need of methods to detect viral pathogens and viral indicators in the point of use to reduce public health risks, preventing outbreaks and increasing the efficiency of humanitarian interventions. We propose a method for detection of viruses in 10-liter water samples, which is divided into 3 steps: viral concentration, nucleic acid extraction, and viral detection. All these steps have been adapted from methods currently in use in our laboratory and can be performed in the point of use by using portable reagents and equipment, reducing the dependence of power supply and freezers. Also, a preservative